Welcome back. So, today we're going to dig into what happens after our body has gone through all the trouble of producing antibodies. You see, it's not just a simple reaction, it's a whole strategic playbook our immune system uses to take down pathogens. Okay, let's dive right in.
So, here's how we're going to break it all down. First, we'll get to know our key players, the antibodies themselves. Then, we'll look at their main defensive strategies, or their plays, if you will. We'll also see how they call for some serious backup, how microbes try to outsmart them, and then we'll wrap up with the absolute must-know essentials for your exams.
Right. So in previous sessions, we've talked about how B cells get activated and start cranking out all these incredible antibody molecules. But the big question is, what do they actually do? I mean, how do they go from just being molecules floating around to actively protecting us? Well, that's exactly what we're going to get into today.
Okay, first things first. Before we get into the fancy plays, let's really understand the mission. What is humoral immunity really all about? And what is it about an antibody's design that makes it so perfect for its job? So, humoral immunity is all about dealing with threats that are outside of our cells. Think of things like bacteria just floating around in your blood, or nasty toxins that they've released. And the weapon of choice here? Secreted antibodies. Their whole job is to hunt down and neutralize these extracellular enemies before they can do any real harm.
And this is where the antibody's genius two-part design really comes into play. You've got the Fab region, which you can think of as the business end. It's like a highly specific grappling hook that latches onto one particular enemy. And then you've got the FC region. This part is like the handle, the part that screams to the rest of the immune system, "Hey, over here, I've caught one, come and get it."
All right, so now that we get that two-part structure, let's see how it translates into action. We're going to walk through the three main plays that antibodies run to keep us safe.
First up, we have the most direct and honestly elegant play in the book. It's called neutralization. It's all about stopping a threat dead in its tracks.
The key to neutralization is that it's all about prevention. Just imagine, a virus is like a key, and it's trying to unlock one of your cells to get inside. Well, a neutralizing antibody just comes along and binds to that key, basically putting a cover on it. Now it can't fit in the lock anymore. It's a simple physical block. And this is exactly why so many vaccines are designed to create a flood of these neutralizing antibodies. Okay, next up is a play that's all about teamwork. It's called opsonization, and it's how antibodies make microbes look way more appealing to the cleanup crews of our immune system.
Yeah, this really says it all, doesn't it? Opsonization is basically tagging for disposal. The antibody acts like a bright, flashing neon sign, making the pathogen super visible and, well, delicious to phagocytes, the cells that are built to eat invaders.
So here's the blow-by-blow. The antibody's Fab region grabs the microbe. This leaves its FC region, that handle we keep talking about, sticking out. Now, phagocytes are covered in receptors that are a perfect match for that FC handle. When they lock on, it's the green light to engulf and completely destroy the microbe. And this is absolutely critical for fighting off certain bacteria that have these slippery protective capsules that would otherwise let them escape.
The third play, well, this one's a bit different. It's a specialized tactic for a pretty grim but absolutely necessary task, taking out our own cells that have been compromised and turned into virus factories.
This whole process is called antibody-dependent cellular cytotoxicity, or ADCC for short. So what happens is an antibody, usually an IgG, finds viral proteins on the surface of one of our own infected cells and latches on. This leaves that FC handle exposed. But this time, a different player shows up to the party, the natural killer, or NK cell. The NK cell locks onto that FC handle, and that's the signal it needs to release a cocktail of lethal granules, killing the infected cell before it can release thousands of new viruses.
Now, antibodies are incredibly powerful, but sometimes you gotta call in the heavy artillery, and that is where the complement system comes in. Think of it as a massive cascade of proteins that works as a major amplifier for the whole immune response.
Okay, this is a really key concept, especially for your exams. Understanding how complement gets switched on. There are a few ways, but they fall into two main groups. You've got the classical pathway, which is part of the adaptive immune response. It gets kicked off by antibodies that have already marked a target. But then you have the alternative and lectin pathways. These guys are part of our innate defenses. They can get triggered directly by a microbe's surface. No antibodies needed. So no matter how it starts, the result is this incredibly powerful cascade. It all begins with a protein called C3 getting cleaved.
This creates a piece called C3b, which is a fantastic opsonin. It just plasters the microbe with eat-me signals for phagocytes. At the same time, other little fragments get released, and they act like a chemical siren, causing inflammation and recruiting more immune cells to the fight. And then, for the grand finale, the cascade builds what's called the membrane attack complex, or MAC. It does exactly what it sounds like. It literally punches holes right through the pathogen's membrane, making it burst and die.
Of course, this is not a one-sided fight. I mean, through millions of years of evolution, microbes have come up with their own clever counterplay book to dodge these defenses. And understanding how they do that is another one of those key objectives for your course.
So let's look at their tactics. A classic move is something called antigenic variation. You see this with viruses like influenza. They're constantly changing their surface proteins, so the antibodies we made last year just don't fit anymore. Other bacteria have figured out how to directly inhibit complement activation, basically shutting down that whole part of our response. And then you have some, like Streptococcus, that wear a disguise. They surround themselves with a hyaluronic acid capsule that looks just like our own tissues, so our immune system just ignores it.
Okay, let's wrap this up and bring it all home. This last part is your quick and dirty study guide. These are the absolute core concepts, the stuff you really need to take away from all this. So let's just run through the essentials one more time. First, remember the antibody's two-part structure, Fab binds, Fc activates. The three main plays are neutralization, opsonization, and ADCC. Complement is that powerful backup that amplifies everything. And for your exams, it's really helpful to remember the special jobs of certain antibodies. IgA is the guardian of our mucosal surfaces, and IgG is the only one that can cross the placenta to protect a newborn. And finally, never ever forget that this is an arms race. Microbes are always evolving to fight back.
And that really leaves us with a final, really crucial question to think about. This constant evolution of pathogens is one of the biggest challenges we face in medicine. As they find new ways to hide from our antibodies and our complement system, it forces us to get smarter. How can we design vaccines and therapies that can maybe anticipate these changes, or even target parts of a pathogen that it can't afford to change? It's a fascinating and incredibly important scientific battle that's happening all the time.